Welcome to the third video in the unit on the adaptive immune system. Previously, we've talked about kind of the general overview of how adaptive immunity works and antigen presentation. Now we're going to start pulling that together to look at how antigen presentation actually activates T cells and what they then do once they're activated. So let's look at T cells and how they function. The adaptive immune response is going to start with activation of helper T cells. So your helper T cells are called helpers because they're the ones that are actually going to conduct the rest of the adaptive immune response. They're the ones that say, this seems to be this type of pathogen, we're going to need this specific response to it. And so often you'll see them written as TH with a 0. Those are called naive helper T cells because they haven't been activated yet. Remember, we already talked about your helper T cells have a CD4 coreceptor, so you'll hear them used interchangeably. Sometimes you'll call them CD4 cells, sometimes you'll hear them called helper T cells. They're the same cell. And that CD4 allows the helper T cells to interact with MHC2 molecules. Remember, those MHC2 were presenting antigen that had been phagocytized, so exogenous antigen from the environment itself. Once the helper T cells become activated, so once the cell comes over and presents to them, they're going to produce a whole bunch of different cytokines. Remember the voices of the immune system that help direct the cells and tell them what to do. And those helper T cells are going to decide, do we need to have Th1 cells? So they'll either differentiate and become what is called a Th1 helper T cell. Those cells will activate the cytotoxic T cells to go do their job. Or do we need to differentiate into a Th2 helper T cell? If so, those cells will form and they'll go and activate B cells for them to do their job. So we're going to walk through the process of how do you activate a helper T cell and then what happens if it goes down the Th1 line or the Th2 line. Okay, so if you think back to when we talked about inflammation, remember we had the tissues out here. It had some sort of disruption that let bacteria come in. And one of the cells that was here where the bacteria were coming in the tissue were the dendritic cells. So those dendritic cells did phagocytosis. They took those bacteria into the phagosome, fused with the lysosome, and then remember what we've just learned is that those dendritic cells, so if we draw that cell bigger, it's going to take that exogenous antigen that it took in through phagocytosis, and it's going to present it out on an MHC2 molecule. Okay, so remember here are our little bacteria. We'll color them in to match. It took in the antigen, and now that antigen is presented out here on the MH2 molecule. This dendritic cell is now going to leave the tissue. It's going to enter the lymph, and it's going to travel to, say, the closest lymph node. Because remember, it's the secondary lymphoid organs like lymph nodes, which are where all of your lymphocytes are. And the key cells that it's looking for are those helper T cells. In the pool of cells, and remember there's going to be a bunch of them, they're all going to have different T cells with different receptors. Okay, so these are all helper T cells. They just all look slightly differently. One of them is going to have the magic T cell receptor that binds to the antigen that's being presented. So remember, this is a helper T cell. This is a naive helper T cell because they're just sitting in the lymph nodes waiting. This is an MHC2 molecule. So remember, we've got a CD4 coreceptor that's interacting with it, which is how we know that these are helper T cells. And upon this interaction, this is going to cause both the dendritic cell and the helper T cell to start producing cytokines. And those cytokines lead to the activation of the helper T cell. And based on what this antigen is and what cytokines are produced, it's going to either differentiate into what we call a Th1 cell or a Th2 cell. Right now, we're going to first look at what happens if it decides to go down this pathway. So here's now our here's our Th1 cell. This pathway happens when this antigen that was phagocytized originally came from an intracellular problem. So an intracellular pathogen, that could be a bacteria that lives inside the cells, or it could be virus, or it could be an intracellular problem like a cancerous cell. But either way, the helper T cell is going to go down the Th1 response if originally this, say, had been a virus, so it would be inside some cells. Right now it's just out in the environment because it hadn't gotten inside another cell yet. When that happens, the Th1 cells are being activated because the cells that we need are going to be those CD8, or what we call the cytotoxic T cells. So at this point, we've activated a helper T cell, and the helper T cell's job is to decide what does the body need and to go off and activate it to make it happen. What helper T cells do is, now that we have a Th1, it's going to produce a different set of cytokines. And those cytokines are going to come up here and tell this dendritic cell, you're going to need to represent that antigen on the other type of MHC molecule. So this dendritic cell pulls this antigen back into the cell itself and instead packages it up and puts it out, instead of on an MHC2, on now an MHC1 molecule. Because remember, any of your body cells all have MHC1 molecules. The MHC2 are unique only to those antigen-presenting cells. So dendritic cells have both. So what we've done here is the Th1 has said you need to switch what you're actually presenting with. So we don't want to present to MHC2 anymore. Now we want to present with the MHC1. What that does is it causes this presentation process to happen all over again, only now it's happening with CD8 cells. So again, remember, we have a whole pool of cells. They all have slightly different T cell receptors, but these now, instead of being helper T cells, are cytotoxic T cells. They all reside in the same lymph nodes. And what's going to happen is the one with the receptor that matches is going to bind. Remember, these are cytotoxic T cells, so its co-receptor is a CD8. And upon binding here, now we're going to produce more cytokines from both the helper T cell and the dendritic cell, which are going to say, you're the winning cell, you need to produce effector cells. Hey, this goes through that clonal expansion, so that now that cytotoxic T cell that matched the antigen can go off into the body and do its job as an effector cell. So this activation of those CD8, the cytotoxic T cells, is really what we call cell-mediated immunity, because it's the activated cytotoxic T cells that are now going to go out into the body and do something. And what their job is to go out and kill any of our own body cells that are currently having a problem. So remember, they were activated by MHC1, which presents antigen that should be inside the cytoplasm of cells. It means there's something wrong internally with our own cells. They're infected with a virus, they're cancerous, and the only way to get rid of them is to actually kill our own body cells with those molecules called perforin and granzymes, these same types of secreted molecules that are used on lots of different cell types. And these punch holes in membranes, and they kill the cell. So now we're going to take a look at how that process works. Okay, so now we have this activated cytotoxic T cell. Okay, remember, it's in the lymph nodes. And it has that specific T cell receptor that was able to interact with that one specific antigen. Out here in the tissue, remember where that inflammation was happening, we have some sort of intracellular problem. That's where the antigen came from to begin with. So say we have a virus, and right now we have a couple of virally infected cells. Those body cells all have MHC1 molecules. And remember, every single one of your body cells has MHC1 on its surface. And so what happens is when the cell itself is infected by this virus, it's going to be putting that antigen out here on the surface. Mainly what it is, it's a big flag that says, hey, I'm infected, someone needs to come do something. The lymphocytes, okay, so your activated cytotoxic T cells leave the lymphatic system, they travel into the tissues, and what they're looking for is these body cells, okay, that match their T cell receptor. Remember, we're also going to have that CD8 co-receptor. So when this interaction happens, this is now telling this T cell that this cell is infected and it needs to die. So the cytotoxic T cell is going to release all of those killer enzymes that it has. Those killer enzymes are going to come here and it's going to destroy that cell before the virus can finish replicating so that the virus can't get out and now it can't infect neighboring cells. For these cells here that didn't have any viral antigen, the cytotoxic T cell won't disturb them because there's nothing there that's being presented to it. However, notice these granzymes that are being released are just released out into the tissue. Typically, they will kill neighboring cells as they diffuse away because they don't just go right to the only infected cell. So this is how cytotoxic T cells are able to help clear intracellular problems when you have viruses that are inside cells. We can't get to the virus directly, but we can kill our own self cell that's making more copies of the virus. So remember, all of this process started because we had a helper T cell that had gotten activated by a dendritic cell that was presenting antigen on an MHC2 to this T cell. And what we just walked through was what would happen if that helper T cell, based on this antigen, differentiated into a Th1 cell, which would then activate your cytotoxic T cell, which is then going to go off into the body to kill any infected self cells. Now we're going to look at what happens if instead, when this antigen is presented, the helper T cell differentiates into a Th2 cell, which is going to lead to the activation of B cells, which are then going to produce antibody for the immune system. So this helper T cell activation of B cells is important because it allows them to produce a really strong antibody response, allows for something called class switching, which we'll look at, and the development of memory cells. So sometimes B cells can be activated without T cells, but it makes a much weaker response, and it's really not how we want the immune system to function. One thing to note as we move into how B cells are activated is to keep in mind that B cells are also considered antigen-presenting cells. So they are able to internalize antigen and present it out on MHC2 molecules. This is going to be important for their interaction with those helper T cells. Okay. We'll then look at what happens when B cells differentiate into what we call plasma cells. Those are the effector cells that are actually producing antibody. And if you look at the two pictures below, you'll see a normal B cell, okay, on the left, and then you'll see what it looks like after it's activated. You'll notice all the endoplasmic reticulum in there. Mainly plasma cells become just these little secretion factories that are pumping out lots and lots and lots of antibodies. So let's see how B cells are actually activated. The activation of the B cells is really a two-step process. So the first, we have to have the dendritic cells or the macrophages come over and activate the initial T cell. So remember again, that dendritic cell is going to present antigen on its MHC2 molecule. It's going to present it to the one helper T cell whose receptor was able to match it. And that's going to then have this helper T cell, in this case, differentiate into a Th2 cell. And in doing so, it's going to go through clonal expansion. So now we have a number of these Th2 cells that all have that same shape receptor. But at the same time, we have something else happening, and that has to do with the B cell pool. So just like there's a bunch of T cells that have different shaped receptors, there's also a bunch of B cells that have different binding sites on their receptors. So for example, we might have some squares and some triangles. And here's one that has a B cell receptor that's binding to those circular antigens. The B cell is actually going to bind the antigen on its own. So remember, T cells couldn't bind antigen, they had to be presented. but B cells can. And when that antigen binds to this specific receptor, this B cell is going to pull that in, so it's going to endocytose it and pull it into the cell, put it into a vesicle, and then it's going to present it back out on the surface. And remember, since it was originally an exogenous antigen, it's going to get presented on MHC2 molecules. So here's two things that have happened. We now have helper T cells that are activated and ready to go, that have a T cell receptor that matches our antigen. And now we have a B cell out of the whole pool of B cells that has bound to the antigen and flagged itself to say, hey, I'm the winning B cell. I'm the one that's going to work. This helper T cell down the 2 line is now going to come over and interact with that B cell that's presenting the antigen. So this is what that would look like. Here's the B cell. It's got that MHC2, which is now loaded with the antigen that had took in from the outside. And it's going to interact with that Th2 cell. Remember, this is a helper T cell still. It's got a CD4 molecule that interacts with the MHC2. This interaction causes the production again of cytokines. Those cytokines are released. They activate the B cell, so the B cell can now go through that clonal expansion. And when it does, we're going to make a whole lot of what we call plasma cells, which will then produce antibodies. At the same time, remember, a few of them are going to form memory cells that will then stay in there for the next time we see a pathogen. In the next video, we'll talk about antibodies and how they function.